Genotypic is very happy to go from genomics, which was looking at high throughput sequencing of DNA and RNA, to now look at a new domain of high throughput protein analysis quantification using the Somalogic platform. This is one of the unique platforms that allows you to look at as high as 11,000 proteins in the human and mouse models, and potentially other species as well, with as little as 50 microliters, and very, very exciting technologies. Thank you, Ji, and thank you, Caroline, for taking time off to be part of this webinar by Somalogic and Genotypic. And over to you, Ji, to introduce the technology and the speaker.
Hello, thank you, Dr. Sudhar. Yeah, very happy to work with Genotypic to bring this webinar to India customers. So my name is Jiang Jie, the Regional Sales Manager, taking care of Southeast Asia, Taiwan, and India market for Somalogic business. So Somalogic is a leader in high-plex proteomics, providing sensitivity, specificity, reproducibility, and scale using aptamer technology to provide advanced biomarker discovery in a variety of therapeutic research areas and drug development. Somalogic has been providing highest plex proteomic solution in academia, biotech, and biopharma market for more than 20 years in US and Europe market. So early this year, Somalogic merged with Standard Biotools and further enhances our proteomics solutions to our end customers. So today, I'm very glad to announce our partnership with Genotypic to bring this high-plex, high-throughput proteomics platform and solutions to India and academia and industry customers. Now, please allow me to introduce our speaker, Dr. Caroline Chen. Dr. Chen is currently the application manager at Somalogic, APAC. Caroline completed her PhD from University of Queensland in developmental biology. After PhD, Caroline has worked multiple international biotech companies, providing application and technical support for customers in APAC region. So welcome, Dr. Caroline, pass to you.
All right, thank you. Thanks for the introduction. Can you hear me?
Yeah.
Okay, great. Let me share my screen. All right, okay. So good afternoon, everyone. Thanks so much for joining the webinar today. All right, okay, full screen?
Yeah, full screen.
Okay, great. So, so in today's webinar, I'm going to go through the introduction to the Somalogic technology. As Jiang Jie has mentioned, we are the high-plex protein profiling a company that provides a high-plex protein profiling solutions for our customer in biomarker discovery, and as well as a lot of other applications with our SomaScan technology. Then on the second part of my talk, I would like to also give some examples of how other customer has been using the SomaScan platform in different applications. All right, so who is Somalogic? So Somalogic has been around for actually more than 20 years, and we have recently merged with the company Standard Biotools. So we are now under the Standard Biotools umbrella, and then as a company, Standard Biotools, we offer both the Standard Biotools platform as well as the Somalogic proteomic platform. So in terms of the Somalogic proteomic platform, we are the currently the highest plex proteomic providers in the industry, where we have recently last year launched our 11K protein detection panel. So in 2015, we started with the 1,500 targets protein detections, and over the years, we have been developing and increasing the number of targets in our SomaScan panel to enable customers to detect as many targets as possible. So now we are offering two panels, both the 7K panel as well as the 11K panel, to enable customers to detect for near 11,000 protein targets within starting materials such as plasma and serum biological metrics. So the key to the Somalogic technology really relies on this reagent known as the SOMAmer reagent, and it stands for slow off rate modified aptamers. As the name implies, SOMAmer reagents, slow off rate modified aptamers means that when this SOMAmer reagent has a very specific binding to its targets, but it has a very slow off rate because the reagent that we selected for in our panels have a very high binding affinity to its target. Then under general or normal conditions, right, when proteins binds to the DNA and they are not very specific binding, they tend to dissociate fairly quickly in the solutions. So with the SOMAmer reagent, these are actually chemically synthesized single-stranded DNA, where we make chemical modifications to this DNA to increase the binding affinity DNA to the proteins. So as you could see from the, let me just show you, disappointing. So as you could see from the image on your right, so the white part is your protein of interest, and then the green and the purple part is the SOMAmer reagent. And the purple region is where we make these chemical modifications on the single-stranded DNA, which mimic amino acid side chains that are known to be involved in protein-protein interactions. So because we synthesize this DNA in-house ourselves, right? So our batch-to-batch reagent consistency is very high. So our current medium CV for our assay using plasma and serum as the validated sample is only at 5%. So when we first started with screening for the SOMAmer reagents, we started with a library of 10 to the power of 15 DNA sequence with these chemical modifications. And then we screen for like purified proteins, either we purchase commercially or we express ourselves, and then screen for the particular SOMAmer reagent that will bind to these specific targets that we have in mind to place in our panel. Then after multiple rounds of this screening, we will come down with a few SOMAmer reagents that will bind to these targets, and then we went on for further validations before we put the final targets in our panel. So here is an illustration that that demonstrates that the DNA aptamers, which is what we use in the SomaScan assay, has very similar binding surface to the antibodies. So in this particular publication, they demonstrated that the binding surface of the DNA aptamers and the antibodies are very similar, which demonstrate that they actually recognize the epitope in a very similar fashion. And one of the advantage of aptamers is that they are 10 times smaller compared to antibodies. The size of the aptamers is around 8 to 15 kilodaltons, while antibodies is about 150 kilodaltons. So in this particular publication, they also demonstrated that the shape complementary index between the antibodies and the aptamers are also fairly similar. So here is a high-level overview of the workflow of the assay. So as you could see, the SOMAmer reagents are in purple. So each of these SOMAmer reagent has got 3 different moieties attached to it. So it has a biotin molecules which enable binding of the SOMAmer reagents on the structure avidin bits. It has a photocleavable linker which enable cleavage of the SOMAmer reagent and the protein complex using the UV light that are placed in our instrument. And then the fluorescence tab which enable detection of the SOMAmer reagent on the readout system, which is on the microarray platform. So upon binding of your samples with the SOMAmer reagent, as shown in image B, right, there will be specific protein binding between the protein and the SOMAmer reagents, and there are also non-specific binding that happens between the SOMAmer reagents and the protein. But as mentioned before, usually when proteins binds to, when non-specific protein binds to the SOMAmer reagents, they dissociate fairly quickly within seconds to a minute. But because our SOMAmer reagent has slow off rate properties, right? So when they bind to their specific targets, they will stick together for a long time. Then the next step is that we then biotinated the protein complex, protein part of this complex after the first binding step. Then we photocleave this protein and DNA complex of the beads. And then during this process, right, during the photocleavage process, we also add another level of specificity as shown in image E here. So we add these poly-N-ionic competitors shown in green. They are also single-stranded DNA where it tends to bind to these non-specific proteins that are released into the solutions where there is non-specific binding that happens between the proteins and the SOMAmer reagents. We then capture this protein and SOMAmer reagent complex back to the beads again. And then in the final step of the workflow, we then release the SOMAmer reagents with the fluorescence tag using a high sort buffer. And what we detect then is this fluorescence tag SOMAmer reagent on the microarray system. So we then hybridize these SOMAmer reagents onto the Agilent microarray, which consists of the probes that is complementary to the SOMAmer reagents. So the SOMAscan assay is a relative quantification assay where basically you are comparing the fold change differences between the groups of sample that you wanted to compare to. So one of the key challenge with proteomic assay is the ability to detect a wide range of protein concentration within biological metrics, such as the human plasma samples. As you could see from the figure on your left, right? So in the human plasma samples, there are highly abundant targets such as the albumin, and then the low abundant targets such as the cytokines. So how do we achieve this wide dynamic range with the SOMAscan assay? So at Somalogic, we have actually run thousands of plasma and serum samples. So we actually understand what are the endogenous protein expression level in the healthy individual plasma serum samples. So we actually optimize our protocol to enable detection of such a wide dynamic range of protein concentration within biological metrics, such as plasma serum samples. So for the plasma serum protocols, the samples will be diluted into 3 different bins, as you could see on the figure on the right. So there will be 1 in 5 dilutions, 1 in 200 dilutions, and 1 in 20,000 dilutions of these samples. And then within each of these dilution bin, the number of SOMAmer reagents are different. So in the least diluted sample bin, right, so there are about 85% of SOMAmer reagents that are targeting these lower abundant targets. And then 12% of our SOMAmer reagents are targeting the mid-range protein targets, and 3% of the SOMAmer reagent are targeting the highly abundant target. So that's how we achieve a 10 logs of dynamic range of the SOMAscan assay to enable detection of this wide expression, protein expression level in the plasma serum material. So the process of the SOMAmer reagent development that we select for the SOMAmer reagent is called the select process, right? So we start with a 10 to the power of 15 random library with random DNA sequence with these chemical modifications. We then screen these purified proteins one by one, right? So we have this 10 to the power of 15 DNA sequence, screen against each of these proteins, and then wash away the unbound proteins, and then elute the SOMAmer reagent that bind to these purified proteins. So we will go through 6 to 10 rounds of this select process, and at the end of each round, at the end of the 6 to 10 rounds, we will then come down to a few SOMAmer reagents that were shown to bind to these specific targets. Then we then go through further validations of our SOMAmer reagents after the select process to make sure that they are of high binding affinity, right? So we will test the K affinity of the SOMAmer reagents. We do a dose response curve to understand the detection of these SOMAmer reagents to the expected protein concentration. We do a pull down assay to make sure that the size of the proteins is indeed the correct size that is expected. And then we also further validate from the pull down assay using mass spec assay to make sure that the proteins is indeed what we have in mind. So because we have run thousands of healthy plasma and serum samples, right? So we also have a pool of these reference populations that we have built with plasma and serum samples, and we have the performance characteristic of each of the SOMAmer reagents in terms of the CV of the SOMAmer reagent, what is the signal to noise ratio in the plasma serum material, what is the LOD of this particular target, the F statistic and the correlation, etc. On top of that, now there are already over 1,000 publications using the SOMAscan technology. So there are also a lot of orthogonal confirmation that have been done by other customers, such as they have done publications using the SOMAscan platform and then has validated the targets with other techniques such as using antibody assay, mass spec assay, gene expression technique, or cis-pQTL analysis. So the 4 primary validations to demonstrate the SOMAmer reagent specificity that we have done in-house, including the determination of the binding affinity of the SOMAmer reagent. We have done the pull-down assay of the SOMAmer reagent with its target proteins, and then we also further validate them with mass spec assay. And then we have done a buffer dose response curve, right, to understand whether the SOMAmer reagent is binding the protein in the expected concentrations, and we also have the performance characteristic of each of these SOMAmer reagents in plasma and serum material. So here is an illustration showing that over 70% of our SOMAmer reagent have more than one additional source of orthogonal conformation. Those could either be our in-house validation assay or additional orthogonal conformation by customers through publications using other techniques such as the antibody conformations, mass spec assay, gene expression assay, etc. We are currently offering two different types of panel, the SomaScan 11K assay as well as the SomaScan 7K assay. As the name implies, with the 11K assay, it allows customer to detect near 11,000 proteins. And then with the 7K assay, it allows customer to detect near 7,000 proteins with just single 55 microliters of plasma or serum starting material. So here is a comparison between our previous 7K and the 11K assay in terms of the content increase in 4 major disease areas in terms of cardiovascular disease, inflammation, oncology, and neurological diseases. We have near 30 to 40% increase in terms of the content in the current 11K menu compared to our 7K menu. And looking at the different biological pathways as indicated in this slide in comparison with our 7K and 11K menu, as well as the nearest competitor, which is an antibody-based assay, we have 30 to 50% content in terms of content compared to our nearest competitor in each of these major biological pathways. So here is the total number of targets that can be detected in both the 7K and the 11K menu. As you could see, the total number of measurements for the 7K is 7,596. So this means that there are 7,596 SOMAmer reagent in the 7K panel. And then for the 11K panel, there are 11,071 SOMAmer reagent in the 11K panel. And in terms of the number of unique human proteins measurements in the 7K menu, it is 6,713 unique proteins. And then for the 11K is 10,070 unique proteins. So in our panel, there are some targets where we have more than one SOMAmer reagent. So here are the multiple measurements to a protein indicates that in the 7K menu, there are about 576 targets where there are more than one SOMAmer reagent. And then with the 11K menu, there are about 706 SOMAmer targets with more than one SOMAmer reagents recognizing this target. And most of the time, the reason for that we have multiple measurements to a single protein is that the SOMAmer reagents are targeting different area of the protein targets. So the easiest way to find out what are available in our current menu is to go to our website, right, menu.somalogic.com, right? So if you do a simple registration with your email and your information, you should be able to access this menu with the full visibility of what is present within our 7K and 11K menu. So as illustrated here, so once you have signed in, you look for 7K or 11K panel and you search for the targets that you're interested, you should be able to see what are the name of the targets, what is the UniPro ID, and then look at the targets that you're interested to screen for. So once you go into each of these targets, you should be able to see what are the target region of the amino acid region that we use for our select process, and then also what is the performance of these particular targets in the human serum and the plasma material. So this is the best way for customers to actually take a look at, for in our menu, what are the targets that are present, as well as are there targets that you're interested and whether they are present in our menu. In terms of the data output, right, so we provide the output file that called the ADAT file. This is basically a text file where you can actually open up this data file in Excel. So basically what you get at the end of the run is a text file that contains each column is the number of the targets that are present in our menu, and then each row is the number of samples that are run in the current study. So what you get is that the RFU, the relative fluorescence unit of each targets in each samples. Then you are able then to compare the fold change differences between the groups of samples that you wanted to make comparison to. Then for each of the run, we will also create a SomaScan quality statement report, in short SQS report. And then in this report, basically, we will let you know the number of samples that you have run, how many samples have a flag, or how many samples passes all the QC criteria of the assay. In terms of the statistical analysis tools, right? So we do have programming packages if customers are familiar with R or Python, right? So they can actually get the programming packages for the data analysis. And then if customers are not so familiar with all these programming language, we also do provide a online web-based tool, statistical analysis tool called the Data Delve Statistical Analysis Tool, where customer can generate an overview of their data by creating such as the volcano plots, as well as the list box plot and the list of targets that are present or highly expressed or significantly expressed within the samples that they are comparing to. So we have also tested some of the animal models on our SomaScan panel, although the initial screening of the SomaMol reagent are based on purified human proteins, but we also have tested some of the animal model plasma serum as indicated in this slide. So we have tested mouse plasma, rat, dog, and hamster, and then look at what are the number of targets that are signaled using the human SomaScan panel. So what we see, for example, in the mouse plasma data sets, we do see 88% of the targets that has been showing signaling in the panel. So, and the observation is that when the amino acid or the sequence identity between, for example, the mouse and the human are very close, then there are higher number of percentage where the targets are also signaling in those samples. So here is a summary of the types of samples that we have run in-house before in our Boulder lab, right? So the core sample types that we have validated or heavily run with our assay are the human plasma and serum samples. As you could see with human plasma and serum, you only need 55 microliters of this study material to be able to detect over 10,000 of protein targets at one time. So although we do request customers to send us 2 aliquot of this studying material, but the actual workflow only required 1 aliquot of 55 microliters of the plasma or serum material. And the reason that we ask for 2 aliquot of this studying material is that if there's any in case there's anything happened during the run, we can immediately repeat the run without having you to resend the samples again. So currently with the 7K menu, right, so all these sample types indicated here, such as the human plasma, serum, urine, CSF, mouse plasma, non-human primate plasma, dog, cat, pig plasma, and other materials such as the cell lysate, tissue homogenates, cell culture condition medium, have been enabled on the 7K menu. And then with the 11K menu, currently we are only opening the 11K menu on human plasma serum material, as well as the mouse and non-human primate plasma serum studying material. So the highlight or the key advantages of the SomaScan assay is obviously the number of plex that we can detect in the studying material with over 10,000 of unique proteins in the 11K menu, which covered about 50% of the human proteome. Our assay or the reagents have high specificity of the SomaMol reagents through our in-house validations, as well as shown in different publications. Then if you are running the plasma or serum material, one of the key advantage of the SomaScan assay is that there's no need for you to have bridging samples between the runs. So let's say, for example, you have run your sample today, and then a year later, you want to add more number of plasma or serum material on the SomaScan assay. There's no need for you to prepare this bridging sample across different runs. And you are able to correlate this data run at different time points together. So the workflow of the assay, right? So you can run up to 680 samples per week, right? If you have one instrument, you can do 2 runs per day or 1 run per day. So if you're doing 2 runs per day, you can run up to 680 samples per week. And then the volume requirement for the assay is only 55 microliters of serum on using serum or plasma material. But of course, if you're running with other sample types, the volume requirement is slightly different, but generally the volume requirement for the study material are quite low. Then our assay has a very high dynamic range with 10 logs of dynamic range to enable customer to detect proteins in the femtomolar to the micromolar range. And very importantly, our assay is very consistent with highly reproducible signal with a medium CV of 5% in the serum and plasma material. So in the second part of my talk, I would like to go through some examples of how customer has been using the SomaScan platform in their studies. So the first example is about trying to gain insight into using protein biomarkers for early cancer detection and cancer stratifications. In this case, the data was generated using the SomaScan 5K panel, which was our previous panel before we have the 7K and 11K. So here they are looking at creating mouse xenograft model from the lung, tumor tissues, breast, colon, as well as ovarian cancer, and then look at the plasma proteome changes at different time points, and hopefully wanted to identify biomarkers that can help to predict tumor volume in this mouse xenograft model. So through their study, they are able to identify 15 blood-based biomarkers that are independent of the tumor type, and these pan-cancer signatures can help to predict tumor volume of certain sizes in the mouse xenograft model. So they started with creating the lung xenograft model. They created 2 mouse xenograft model with the lung tumor, right? So a drug-sensitive cell line as well as the drug-resistant cell line, and then look at the protein expression profile at different time points of this mouse xenograft model and identified 98 differentially abundant targets that are shared between these 2 lung xenograft model. They then used machine learning and created a model and identified 19 proteins that are common between these 2 lung xenograft model that are able to identify the tumor-bearing mice versus the non-tumor-bearing mice, and also the tumor volume prediction. They then further extended their study by creating additional xenograft model by transplanting the ovarian tumor cells, colon, as well as breast cancer tumor cells to create 2 different xenograft models for each of these different tumor types, and then identified 15 common biomarkers that can help to predict tumor volume in these 4 different tumor types. So looking at the pathway analysis in these 4 different xenograft model, as you could see, the number of the enriched pathways as well as the number of differentially expressed targets are different between the each of these xenograft model, with the ovarian xenograft model having the largest number of enriched pathways as well as the number of differentially expressed targets. And then in the ovarian xenograft mouse model, the enriched pathway are focused on the small molecule metabolism, immune response, translational regulation, as well as RNA splicing. While in the breast xenograft model, these enriched pathway are concentrated on small molecule metabolism, proteolysis, immune response, complement cascade. And in the lung xenograft model, they are focused on the cell migration, adhesion, proteolysis, extracellular matrix, with the colon xenograft model having the least number of enriched pathway, and they are focused on the translational regulations, RNA splicing, processing, as well as proteolysis. So this publication demonstrates the potential use of the protein biomarkers in early cancer detection, cancer progression, as well as cancer stratifications, trying to identify signatures that can help to predict tumor volume in the mouse xenograft model. So the next example is about trying to look at proteomic profile differences, and then hopefully to identify organ aging signatures using the SomaScan assay. So this is a fairly recent publication late last year in Nature by Tony Weiss, right? So the aim of this study is to make use of the SomaScan assay to look at the blood plasma protein expression differences across 11 major organs to try to identify organ-specific signatures in these 11 major organs, and can help to predict health as well as disease in living individuals. So the key to this particular publication is that they demonstrated that they are able to combine data generated with different versions of the SomaScan assay. So in this particular study, right, they actually analyzed data from 5 different cohorts. 2 of the cohorts actually run on the 5K assay, while 3 of the cohorts actually run on the 7K SomaScan assay. So they demonstrated that they are able to combine this data together and analyze them together and identify signatures that are independent, that are specific to these 11 major organs. And the total number of individuals that they have analyzed are about slightly over 5,600 individuals. And then their algorithm actually predict this organ aging reproducibly in these 11 major organs. So this particular publication firstly demonstrated that what I've mentioned previously, right, with the SomaScan assay, right, run using the plasma or serum studying material, you do not need to have any bridging samples across the run at different time points, and you are able to correlate your data with these different versions of the assay. And it also demonstrated that they are able to identify protein biomarkers that can help to predict organ aging in living individuals. So the next example is about trying to look at identified biomarkers for Alzheimer's disease. As we know that Alzheimer's disease is a very important neurodegenerative disease due to misfolded proteins in the brain. Traditionally, the ELISA technique has been used to look for biomarkers in Alzheimer's disease, but one of the limitations is obviously the number of targets that you can identify. So then the aim of this particular study, this is actually a follow-up studies of the previous study where they have already identified 5 proteins associated with Alzheimer's disease. And in these particular publications, they wanted to actually look at the comparison between different platforms. So they actually look at a comparison between the SomaScan assay as well as the Olink antibody assay, as well as the depleted mass spec assay using CSF as well and plasma as the starting material. So here on the left is the Venn diagram showing the number of targets identified in each of these different platforms, right, using CSF and plasma as the starting material. So as you could see that the SomaScan assay are able to detect the highest number of proteins in both plasma and CSF compared to the antibody platform as well as the mass spec platform. On your right is the correlation plot between the different techniques, right? So Olink assay compared to the SomaScan assay in both CSF samples as well as the plasma samples. And you can see that the correlation in these 2 different studying material is around 0.69 to 0.7 with the Olink assay comparison to the SomaScan platform. And then if you're comparing the mass spec platform with the SomaScan platform in the CSF and the plasma studying material, the correlation is about 0.62 and 0.7. So looking at the number of positively or negatively regulated proteins in the CSF as well as the plasma material, comparison between the 3 different platforms, again here it demonstrated that the SomaScan assay are able to detect the highest number of positively or negatively regulated proteins in the Alzheimer's disease patients compared to the other 2 techniques. And on the right is the Venn diagram showing the number of significant targets identified in the different platforms in both the CSF as well as the plasma, whether they are positively regulated or negatively regulated. So this study shows that, the SomaScan platform are able to provide a deeper coverage of brain-related protein network compared to the mass spec technique as well as the Olink assay. And then many of these targets identified with the SomaScan assay also matches with the traditional measurements such as the tau measurement as well as the Montreal Cognitive Measurement Score. So this study provide a comprehensive evaluation of the different methods for protein detections, right, using two different sample types, CSF as well as plasma, and demonstrated the SomaScan assay was able to detect the highest number of proteins detected in both the plasma and CSF compared to the other two techniques. And then many of these targets that are identified with the SomaScan assay has also highly correlated with standard measurements such as the tau measurements and the Montreal Cognitive Measurement Scores. And then in this particular publication, the author actually mentioned that one of the advantage to them with the SomaScan assay is that for certain proteins, as I mentioned, there are multiple SOMAmer agents against the same target, and then this enabled them to actually discover new biology. So the next example is about trying to identify biomarkers in colorectal cancer. And in this case, right, the studying material is actually using stool samples from colorectal cancer patients versus the healthy control. This was done with the very early version of our assay, which is the 1.3K assay. So the aim is to try to identify biomarkers for colorectal cancer, because we know that colorectal cancer is a very common cancer. And then if you're able to detect this cancer in the very early stage, there are higher chances for you to recover from this cancer. So they wanted to try to identify biomarkers in colorectal cancer and hopefully can help to improve some of the screening tests. For example, there are risks involved with colonoscopy. So they collected stool samples from the colorectal cancer patients as well as healthy control, run it on the SomaScan 1.3K panel, and then look at what are the protein expression differences between the colorectal cancer patients versus the healthy control. So on your right are the top 50 protein biomarkers that they identified between the colorectal cancer patients in red versus the healthy control. They then validated their SomaScan data with the antibody ELISA platform, right? So on your left are the top 16 targets that they validated with the ELISA assay that show high concordance between the SomaScan assay and the ELISA assay. So this is a demonstration that the SomaScan assay, although was very heavily validated using the plasma and serum studying material, but it also works with dirty samples such as stool material. And then in this particular publication, they are able to identify biomarkers in the colorectal cancer, and then they also validated that with the ELISA assay and show that the SomaScan data show high positive correlation with traditional ELISA methods. So the next example is about trying to actually develop a LDT test with the SomaScan assay. So OncoHost is actually a company that have the platform to run the SomaScan assay. They do have the instrument in their lab, and they developed this test called the Prophet platform. So using machine learning, they developed a algorithm that can help to predict, give better information to clinicians to make better decisions whether the patients should go on certain treatments. So this Prophet assay is a LDT test that they have developed based on the SomaScan platform and model development that help them to enable clinicians to give better information to patients' treatment. So they have actually published their work regarding to the validations of their Prophet assay. So I will not go through details on the assay development. But what I wanted to point out in these particular publications is that they have demonstrated our SomaScan assay precision as well as accuracy. So on your left is the experimental precisions, where they have two different technicians running the SomaScan assay and then looking at the intraplate CV as well as the interplate CV of the SomaScan assay. So in their hand, they also demonstrated that the SomaScan assay has a very low medium CV of around 4 to 6%, which demonstrated again this low CV of the SomaScan assay is not only producible from Somalogic itself, but it is also actually reproducible in customer hand. And on your right is the demonstration of the experimental accuracy of the SomaScan assay, where they actually send the same samples run in their lab to our Boulder lab to run, and demonstrated highly correlations between the two sites. They then also look at the computational precisions of their readout methods, right, of their results, whether their algorithm is accurate in terms of the predictions, right? So they run multiple samples with many replicates and then look at the predictions. So they demonstrated that 93% of the samples show 100% precisions in terms of the readout. So this is a demonstration that the SomaScan assay is not only useful in terms of the research where enable customer to look for biomarker discovery. It can potentially be developed into, in this case, a LDT test, which is a test that help clinicians to make better decisions of whether the patients should go on a chemotherapy itself or chemotherapy with immunotherapy, and then this enable personalized medicines in the future. So the last slide, I would like to show or demonstrate that the SomaScan platform has also been utilized in many large biobank projects and cohort studies. So here is an example of the large cohorts that have been run on the SomaScan platform, right? For example, last year we have run 4,000 samples in the China Kadoorie Biobank. With the EPIC study, we have run like 30,000 samples with the EPIC studies, as well as 15,000 samples with the MESA study, generated millions of protein measurements to enable customers not to just look at fundamental discoveries, but combining with their genetic data as well as lifestyle clinical data to enable customers to gain better insights into the biology and in the disease that they are interested to investigate. So bottom of the slide show additional biobanks or cohort studies that have made use of the SomaScan platform. Then this slide is trying to demonstrate again regarding the ability to actually combine your data together, right? If you are running your samples at different time points, you are able to combine the data together without having any bridging sample. As for example, illustration on the first study, right? So with the ERIC Visit 5 study, they had run it in 2018, and then with the ERIC Visit 3 study, it was run in 2019, and the BLSA cohort was run in 2021. So they are able to combine these three different cohorts together and then look at dementia prediction. So this is a illustration that, the SomaScan assay, right? If you're running like plasma and serum material, you are able to combine the different data at different time points together without having any bridging samples in the different run. And again, a demonstration that even if you use different version of assays, you are also able to combine the data together for analysis. So with that, actually, that's the end of my presentation.